Yeah, so hello and thank you to everyone who came out to see my talk. I'm very excited to be presenting some of my unpublished work on the application of human kinematic parameters for measuring motion performance in mice. So a common assay that's used to study motor learning in rodents is called the Single Pellet Grasping Assay. In the Single Pellet Grasping Assay, mice are food restricted to increase motivation to reach for a high reward food pellet. You can see here in the reach panel, the mouse or the rat will begin by lifting its paw, advancing forward, pronating its digits, and then grasping the pellet. And once the pellet is grasped, the paw then returns the pellet back to the mouth. What's interesting is this motion seems to be relatively conserved between humans during a reaching and grasping task. So if you look at the hand here shown in a non-visual reaching task for the human, it starts, lifts, advances, pronates, and then grasps the object, very similar in a fashion to the mouse. These findings, along with more, have made it a good model for studying the mechanisms of motor learning as well as execution, as research supports that there is a developmental homology between the two. Traditionally, motor efficiency in rodents can be quantified by looking at simple ballistic parameters such as velocity, angular velocity, or even just the percent of success during retrieval. Qualitatively, this can be characterized using different subjective scoring systems, such as the Eskol-Wachman movement notation. An issue that occurs here is that subjective analysis of motor performance can introduce bias as well as variability in what one observer quantifies as an efficient or an inefficient motion. A common objective measurement for quantifying motor failure is needed in order for us to improve our understanding of motor learning and understand how a more efficient motion is developed over time with experience. So in humans, a common use parameter to measure motor learning and efficiency is called peaks, or also known as submovements or intermittency. So here, peaks are defined as any change in acceleration or deceleration in a trajectory are such that acceleration is equal to 0. When peaks are increased in a movement, this is typically indicative of a less efficient motion. Another parameter that is used in quantifying human parameters is something called mean squared jerk, or jerk for short, which measures the smoothness of motion. And this is taken by looking at the area under the curve of the integral of the acceleration. A study by Rand et al. Showed that Parkinson patients have increased movement duration as well as increase in intermittency, or what we would quantify as peaks, during a reaching and grasping task. So you can see they take longer, as well as if we look at acceleration compared to human, there's a lot more fluctuation in this, suggesting that they have a less efficient motion. While these parameters have been well established in humans and primate models, we wondered if this could also be true for mice. We're interested in modeling these parameters in skilled reaching due to the wide variety of different genetic and molecular tools that can allow us to probe the underlying circuitry behind the behavior. Particularly, our lab is interested in understanding skilled reaching and grasping in a female mouse model of Rett syndrome. So in collaboration with Dr. Eric Wade, who was a previous engineer at the UTK Engineering School, we sought to answer if these known kinematic parameters in humans, such as peaks and MSJ, could also be applied to mice. We hypothesized if these patterns of motor output during a reaching and grasping task are relatively homologous between humans and mice, then we would expect that known parameters such as peaks and MSJ would be able to predict motor efficiency in mice. So we asked the questions, are there kinematic differences in successful reaching versus failed reaching in the single pellet grasping assay, specifically looking at changes in mean squared jerk and peaks over time, as well as is there any correlation between the number of peaks or the average MSJ in terms of percent success rate? If peaks or MSJ are predictive measures, then these should decrease over time with training and could build a predictive model for motor efficiency in mice. To answer these questions, we used the single pellet grasping assay with 2-month-old female wild-type mice and restricted them to about 10% of their body weight and maintained this. And then we began motor training. So during the motor training, we allowed mice to reach for as many pellets as they would like by reaching through a slit in their chamber, and recorded the number of successes and attempts during this process, while simultaneously recording the behavior using high frame per speed video recording. These videos were then inputted into a video analysis software called Tracker, which would then allow us to manually track the paw over time, and then later on derive some of these kinematics that we're interested in. The data that I'm going to show you is an N of 428, meaning that I tracked the reaches for these mice for 428 individual reaches, and this took a while. So here I'm showing you a live demo of what we see in the Tracker motion analysis software, and this is a really cool video, right, where you see the cyan dot where I'm tracking the paw manually in this software, and it's bringing it back to its mouth. In the right panel, you can see that there's this change in X over time where we're getting the X and Y position relative to time. And once we have this data, we can then derive kinematics such as MSJ and peaks using a custom MATLAB script that Dr. Eric Wade at Cal Poly and I designed together. Here I am showing you the average number of peaks for successful reaches in black and failed reaches in red. And all of the data is normalized to the first day of success. So you're looking at fold differences between success and fail. And what we see is that there's a significant fold increase in failed reaches in terms of peaks, and that this is consistent across training. However, as we continue with training around day 4, there is no significant change in peaks. So this suggests to us that it's possible that the failed reaches are becoming more mechanically similar to the successful reaches, meaning that efficiency overall is increasing. To further this analysis, we did a linear regression analysis to look at the percent fail and the number of failed peaks during each of those reaches. So these are average per mouse on each day. And what you can see is that there was a positive correlation in that as percent failure increases, the number of peaks also increases. Keep in mind that as peaks increase, this is usually indicative of a less efficient motion and suggests that mice that have higher peaks in reaching usually aren't efficient at the task. And so overall, we showed that this was correlated and that there seems to be a more efficient strategy that is developing over time. So when we look at the other parameter, MSJ, we kind of saw the opposite effect. There actually seemed to be no significant change in MSJ, whether it was a successful reach shown in black or a failed reach shown in red, also normalized to the first day of success. And if we do a linear regression analysis of this, we see that there's virtually no correlation at all with MSJ during this single pellet grasping assay. So in summary, I hope to have shown you that peaks seem to be a predictive measure for motor efficiency in mice, as we've seen that peaks in failed reaching decrease significantly with training, indicating that a more efficient reaching strategy may become developed over time as they become more mechanically similar to the successful reaches. In the context of MSJ, there didn't seem to be any predictive power in terms of motor efficiency. This was surprising since peaks in MSJ are typically dependent on one another. We hypothesized that this could be due to the fact that previous studies looking at MSJ as a quantitative measure have a consistent start point. However, we do this in free-moving mice, therefore the starting position of the paw is different for each reach. My future directions right now are determining if more simplistic measurements such as peak velocity or the time to peak velocity change with training, because this has been previously reported in developmental studies in humans as well as some studies in mice. So that will be important for us to know if those are also changing consistently as well. And so I told you before that we were manually tracking these mice. And at the time when I started this, DeepLabCut really hadn't kicked off. However, thanks to DeepLabCut and Dr. Mackenzie Mathis and the DeepLabCut team, we're now using this approach to quantify motor behavior using automatic techniques through DLC. So here you can just see a preview glimpse of where we're going with this, which is to use the automatic tracking to generate pose estimation data. With that pose estimation data, we can then input that data into our own MATLAB script and then compare that to our manual calculations, which will give us a good quality of assurance for our data. And so with that, I would like to thank my lab, my mentor, Dr. Keerthi Krishnan, Billy, a postdoc in the lab, Dr. Dana Leo, a graduate student in the lab. They've all been implemental in this project, as well as 2 undergrads who helped kickstart this project, as well as our very successful collaboration with Dr. Eric Wade. It's been a journey. So, and with that, I'll open the floor for questions.
Thank you, Mike. Perhaps you may want to get off the share screen there. There we go. Okay. Yeah. So any questions for Mike? Normally, I would be the one asking questions, but then that would become a lab meeting. So let's, if other panelists or anybody else have questions about this, please feel free to ask. Not much at this point. So perhaps we can come back to Mike after this part and move on to our, oh, we may have one. There we go. All right, we do have one. The question is, nice talk. What are the future directions?
Yeah, so right now we're just working on a short manuscript for submission at the Journal of Neuro Rehabilitation and Repair. Ultimately, what we'd like to do is look at this in a mouse model of Rett syndrome. So patients with Rett syndrome have mild to severe sensory and motor impairments. So we can use our mouse model to see if pre-symptomatic versus symptomatic changes in motor efficiency are present in our mouse model. And this could give any type of indication in terms of designing future rehabilitation strategies, as well as probing underlying circuitry later on.
That is correct. Mike's got his work cut out for him. So, all right. Okay, so we'll move on. Thank you again, Mike. That was lovely.
Thank you.
And we move on to our next speaker.